Now I will hand over I will call all the speakers of this session. Dr. Naveen Kumar, sir, please. Then second is Dr. Pragya Yadav. She is there. Our third speaker of this session is Dr. Nivedita. Yes. So I am going to hand over the mic to our chairpersons now. Sir, please. Okay. I will first introduce our chairpersons. Dr. Salunke sir, he is a member in Preventive and Social Medicine. He is a postgraduate, and from 1994 to 2000, he has served as an adjunct associate professor at Biological Sciences, Human Ecology at University of Texas, USA. He has experience of over 30 years. He is a regular contributor to public health development as a member of various state committee and Ministry of Health and Family Welfare, Government of India. He is a regional advisor at WHO in 2005, assistant regional director in 2009, and WHO representative to Indonesia for 3 years. His contributions are demonstrated innovative approaches in public health, assisted state governments in initiating the schemes focused on improving health services for poor sections of the society, actively involved in formulating health system development project for Maharashtra state supported by World Bank. He in HIV/AIDS control program, his contribution is there in assistance with Maharashtra state. It's a part of team designing for NACO phase 2. Current role, he is a senior advisor to the President, Public Health Foundation of India, focusing on health system issues since May 15, 2012. Principal investigator of several public health projects implemented through the Public Health Foundation of India. Due to our second chairperson, is Dr. Jeyakumari. He is a professor and head, JIPMER, Karaikal. His experience is 25 years teaching as well as research. Her area of interest are antimicrobial resistance and infection control. She had 50 various national and international publications. She is a life member of IMM, Indian Association of Medical Mycology, postdoctoral member of American Society of Microbiology, currently executive member of IMM Pondicherry chapter, and she is an external expert for PhD students. Thank you. I am now hand over the mic to Dr. Salunke sir.
Good evening, everyone. It's a privilege to be here to share this session on One Health approach along with Dr. Subhash. So in today's interconnected world, addressing the health challenges requires a holistic perspective, that is integration of human health as well as the animal and environmental health. And it is very evident after this COVID pandemic, we tackled that pandemics as well as we are facing AMR, that is antimicrobial resistance are arising. And this made us to realize the importance of One Health approach. So this approach should not be a theoretical, it should be a practical approach to solve the global issues. And we have distinguished experts here to share their insights and experiences. So without much delay, I call upon the first speaker. Dr. Naveen Kumar, who is the director of the National Institute of Virology in Pune. So, Dr. So after completing his PhD, he studied the persistence of the FMD virus in cattle in Atlanta, USA. He worked on understanding the interactions of the influenza virus with host cell signaling pathways. He is a renowned virologist who made significant contributions to the field of virus-host interactions and epigenetics. He is a pioneer in analyzing drug resistant against host-directed antiviral agents and elucidating the molecular mechanism involved. Due to his extensive publication record, he is considered one of the top researchers in this field. He was the first to isolate the lumpy skin disease virus in India and developed India's first lumpy skin disease vaccine, which has been commercialized. The vaccine is much safer and most efficacious compared to the other LSD vaccines used globally. He has received several national and international awards and is a life member of many scientific societies. He has authored over 130 scientific publications. Welcome you, sir. I hand over the mic to this, Dr. Naveen.
Thank you. And thanks to the organizers. So the ICMR is actually taking the World Health issue in a big way, and I think Dr. Nivedita Gupta will be happy that the theme is percolating very well in each sector in our country. So as already been introduced, so I am going to talk about the lumpy skin disease vaccine. So this virus was actually almost entered little before than COVID-19, and it created a more horrible situation in the animal industry because the mortality was much, much higher as compared to the COVID-19. So we were able to actually develop a very effective live attenuated vaccine well in time. So although I'm working as a director at NIV, but the work was actually carried out at this institute, that the National Center for Veterinary Type Cultures in Hisar. So lumpy skin disease virus, it was restricted for many, many decades to the Africa. So in the beginning of 21st century, it started, moving outside Africa, and then initially in Israel, Jordan, Egypt, and then the Middle East. And suddenly in 2019, it spread to many Asian countries, including India. So this is a pox virus that belongs to the Capripox virus genus, although the two other viruses, the sheeppox virus and goatpox virus, along with this makes this genus Capripox virus. So as I told you, this leads to high mortality, particularly in 2022, because although the virus was first identified in the Odisha in 2019, but due to some COVID restrictions, it was restricted into the eastern belt. But in 2022, it created havoc, particularly in the northwestern part of the country, Rajasthan, Gujarat, Punjab, Uttar Pradesh. So now when there was, lot of outbreaks, there were going on, particularly in the summer rainy season in 2022. So there was one paper that was circulating in the social media suggesting the very severe lesion in a boy. So then there was some, the public health consequences it was considered, but actually that was something else, they could not confirm. So I went after, that paper make a lot of nuisance, and then we went to the animal healthers that were actually closely associated with the carcasses, and they were in gosalas feeding and watering to the animals. So we collected a lot of serum samples, but none of them were found to be positive against antibodies in the human. So beside this, and also no other the association of the virus actually, or the viral genome could be found. But surprisingly, these are the three animal species which have never been shown to infect by LSDV. So that we were first time, show when the extensive outbreaks were there. That was the camel, the horse, and the deer. So beside the anti-LSDV antibodies, we also demonstrated the virus isolation and the viral genome. So if you look at back the impact of the LSD, so one study carried out in Ludhiana, so it suggests that over 18,000 crore rupees just in 2022, and more than 26% milk reduction in the affected animals. So it completely devastated the livestock economy, and some of the gosalas, they completely vanished. All the animals, they died. Some of the small farmers, like managing 10, 20 animals, they know they're just jobless, you know. So it actually threatens the livelihood of the poor and marginal farmers. So the vaccine, the story begins from this letter which we got from the Ranchi, Jharkhand, from a veterinary college. So they invited us to, examine, because this disease was new, people, they were not knowing. So I went there in 2019 in December, so collected the samples, and then we isolated the virus. So we were the first to isolate this virus, and then we characterized it by whole genome sequencing, and we observed that this is a Kenyan type virus which is circulating in our country. So that next objective was, actually we never planned to develop a vaccine, but because this was lockdown, so it was just fun. We were trying to propagate the virus, because the virus was initially isolated in the primary cells, but then my objective was to adapt into the established cells, so that we can, if want to do further studies in terms of the antiviral screening and other things, so we can use it. So initially it was very difficult, For first passage we gave in the Vero cells, it took 15 days. And we couldn't see any evidence of cytopathic effect. But after fourth passage, we see some evidence. So initially it was taking more than 10 to 15 days for to show something in the cells. But after 50 passage, you can see that the virus was producing very clear CP within 48 hours. So after 50 passage in Vero cell, that took us about more than a year. So the virus was very replicating efficiently, growing at a titer of more than 10 to the power 7. And on the whole genome sequences of each 10th passage, we observed that it has acquired most of the mutation had been seen in the other capripox virus vaccines. And most importantly, with this our strain, it has 801 base pair deletion in the ITR region. So that was something special. So we characterized this virus at P50, passage 50 level. We did whole genome sequencing. We also look at it's growing well over the titer of 10 to the power 70 CID50, it's mycoplasma free, and it is non-toxic to the laboratory animals, it's producing antibodies. So at this stage, we actually patented it, this vaccine candidate. And next step, we did the challenge experiment, efficacy studies in the cows. So because the cattle is the major animal species being affected by this virus. So the OIE, or the World Organization of Animal Health, they suggest this protocol that you immunize 10 animals, 5 as a control, then do the vaccination, and 1 month after vaccination do the challenge. So you can see that if you do the infection by intradermal intravenous route, so after 7, 8 days, it produces the local lesions and generalized lesions as well, besides the viremia and the swelling of the lymph node. So this is the outcome of the challenge experiment. So left panel you can see is that these are the immunized animal, vaccinated animals with P50 virus, and right side this is their control animals. So most prominent criteria of if developing a disease is that the animal, the challenge control animals, they should develop the poke-like lesions, at least journal, some local, but sometimes journal as well, and there should be viremia. So you can see that the red boxes indicate that all these critical parameters, they develop the skin nodules, inflammatory reaction, generalized nodule, fever and viremia. But in contrast, you can see on the left side of the vaccinated animal, they were nothing, no reaction, they were absolutely fine. So this suggests that this vaccine is efficacious against the virulent LSDV challenge. So regarding the immunogenicity, besides the experimental infection, we also went back to the field and immunized the animals. So the antibody response is a problem, the LSD, which I am going to talk later. So in experimental infection, out of the 10 animals, they develop, 9 animals, they develop the antibodies. When we went to the field, immunized thousands of animals, about 85% of them, they develop the antibodies. But of course, there was they produced a very nice cell-mediated immune response. Primarily CD8 specific response. We didn't see much difference in the CD4 cells. So the CD8 cells that were activated they were very well able to actually eliminate the virus. Beside, we also see a very nice interferon gamma response when we stimulated the PBMCs isolated from the immunized animals. So this is about the field studies. So the vaccine, the experimental vaccine was distributed to the field, and all the observations were taken. These are some of the data. So if you are trying to look at the efficacy of or safety of the animal vaccine, then you have to also critically, examine the production parameter, particularly the milk. So we didn't see any obvious negative effect on the milk yield. So the virus was not excreted in any the excretion route, nasal, fecal, and oral routes. So one of the limitation of the goatpox vaccine that was actually already licensed for sheep and goat, so it can't be used in pregnant animals. But we observed that this animal, we immunized more than 3,000 pregnant animals of all stages of pregnancy, they were all fine. So, so the vaccine was absolutely safe in terms of the use in cattle. So then, one of the limitation of the live attenuated vaccine, particularly the pox vaccine, which is already being licensed, except the vaccine which we develop, is that sometimes there is a local reaction, and in very few occasions there is small micronodules all across the body surface. So that's why farmers doesn't willing to take the vaccine. And when there is outbreak situation, then what happens? It is very difficult to take a decision whether or not to immunize the animals, in contact susceptible animals. So what happens then? Sometimes you immunize animals who are already under incubation period, and farmers blame us that this disease has happened because of the vaccination. So that was immediately what we did is, as I told you, that we had a 801 base pair deletion, which is never being reported in any vaccine. So we developed a simple PCR by which you can actually very well differentiate the vaccine and field strain. And beside that, it's very useful to ascertaining the purity of the vaccine. So this was also, we have already patented. So then another, the parameter for a vaccine, if you want to run a successful vaccine program, and at the end of each successful program, so if you want to apply to the WHO or World Organization for Animal Health that we are free from the disease, then you have to show that the freedom from the infection. So because we are using the vaccination, so you have to discriminate that the antibodies which are there in the population is not due to the infection, it's due to the vaccination. So one of the limitations with all the LSD vaccine, which is all across the globe being used, is that they don't have a DIAVA concept. So, but for us, the good thing was that there is a major 801 base pair deletion in the ITR region. So what we did is, we cloned that this region. Fortunately, this was immunogenic. So we purify that and use the purified antigen as a tapping antigen to develop ELISA. So you can see that it very nicely reacted with the infected animals, but not the vaccinated animals. So this has already been recently we patented. So this test, this make the DIVA compatibility. This is actually much more important, making this as a very good candidate for the Make in India campaign. So then one of the question is that some of the LSD strain there are, more virulent, some less virulent. Whether or not the vaccine is being developed is going to protect against the remaining strains. So I had a repository of all these strains starting from 2019, and we did a cross-neutralization assays. So it perfectly cross-neutralized all the strain sera that were raised against the vaccine strain. So the vaccine technology was actually released by the Union Minister of Agriculture and the Union Minister of the Animal Husbandry on August 10, 2022. So it was actually released at the time when there were extensive outbreaks were going. So I was asked that how much vaccine you can produce. So, but our capacity, being a lab, so we said that we can maximum produce maybe few lakhs dose. But then they said we immediately transfer the technology. So Within few weeks, actually, we then transferred the technology to the industry. So the 4 major vaccine manufacturers, they came forward to take the technology. That is Biovet Private Limited, is the Bharat Biotech, Hester Biosciences, Ahmedabad, Indian Immunologicals Limited, Hyderabad, and the Institute of Veterinary Biologicals is a government vaccine manufacturer here in Pune itself. So in the history of ICR, this was sold out in a maximum, generating revenue 81 lakhs each. So that 4.8 crore rupees came to my institute. So then actually this is something, the regulatory approvals take times in our country. We were lucky that during the COVID-19 it was quick approval. So we released the technology in 2022, but the CDESCO gave the approval this year, just few months back. But now is everything okay, the product is in the market. So then, as I told you that in 2021, the CPOX and goatpox virus is already there in our country. So lumpy skin disease virus is in the same genus, so they give some sort of cross protection. So based on the literature that it may provide some cross protection, so the government policy makers, the Animal Husbandry Department, they authorized the use of the goatpox vaccine, but it was never tested whether in real sense is going to provide protection or not. But later on, it was observed that even in Maharashtra, all the animals they immunized with the goatpox vaccine, and then there is 32,000 death of the cattle, even those that were vaccinated with the goatpox vaccine. Then our studies, we demonstrated that yes, of course, the goatpox vaccine may induce some degree of antibodies, but it fails to elicit any cell-mediated immune response. So that's why now anyway the original homologous live attenuated vaccine is already there. So now one of the problem with the LSDV is that it's very least studied. So as I told you, the virus has been for many decades it was in Africa. So the lot of studies couldn't take place. So if you immunize the animals with the vaccine, so in field condition 30, 40, 50, 60, 70, 80%, no more than that, animals they develop the antibodies. But if you want to challenge the animals that are free from the LSD antibodies that were vaccinated and want to challenge them, either resist the challenge infection. So that is something, if you want to, in a manufacturer setting, once you establish its efficacy, so most of the vaccine, whatever you do is, a batch is taken, it's immunized, and after 1 month or 3 weeks, you see the antibody titer and pass the batch. But for LSD, this is not the case, because only 60, 70, maybe 80% animals, they are going to develop antibodies. So because this was the new disease, there is no protocols globally. So it was difficult for us to convince the regulators as well. So then, but for me it was possible because I can demonstrate the cell-mediated immune response. But for manufacturers it's very difficult. They can't do the challenge infection again and again. So what we did is, because I could face this question in media, in a lot of platforms, then I decided to do something at least. So what I did is, I did the profiling of the microRNA in primary lamb testicle cells at different time post-infection. I know that such kind of studies doesn't lead to any firm conclusion, because at the end what you are going to get is lot of the genes that are upregulated or downregulated. But for us, we were lucky that we were pinpoint a particular microRNA that were found to serve as a very good biomarker. So we identified a lot of the microRNA that were either upregulated or downregulated. Out of that, we again verify them by the PCR and then tested them, the animals that were experimentally challenged. So finally, we were come up with a very good microRNA, that is microRNA 29A. So this microRNA, if you give the experimental infection, so this, the lesions start developing at 7 or 8 days. But if you look at the microRNA 29 at 3 days, the level goes down. At the same time, the interferon responses are higher. So with a series of multiple experiments, we demonstrated that yes, the microRNA can serve as a very good marker. Early diagnosis of disease even before the development of the clinical science. Besides serving as the biomarker, we further did lot of analysis and demonstrated that if the microRNA level goes down more than 60% in the animals that were vaccinated, and we are looking the microRNA 29 in the PBMCs, so that very well correlates with the protection. So the good thing of this is that now the manufacturers, they do not need to challenge, they can use this data, if the microRNA, you immunize the animal, 1 month later you take the PBMC and then stimulate the LSDV antigen, look the level of the microRNA. If it goes down, particularly for those animals that do not produce the antibodies. So this has also been patented and published. So now another question was that in some of the areas disease was very severe. Some of the farm, they completely vanished. I have seen that some of the Gosalas, 100% animal, they died. But some of the farm, they resisted the infection. The morbidity was very low, 10, 20, 30%. So these were the data one of the veterinarians sent me, that I have seen that the disease is significantly higher morbidity and mortality in the crossbred cattle as compared to the Rathi cattle. So as a scientist, I said I should do something, because this question was faced by me again and again at several places. So what I did is, I went to a farm where there is a disease 6 months ago and screened them. There were antibodies there, and then collected the PBMCs and then stimulated with the LSDV antigen. And we observed that the Rathi breed of the animal, which has a very significantly lower morbidity and mortality, they actually mount a more potent cell-mediated immune response, which is manifested by the low microRNA level and higher interferon CD8 response or cell proliferation responses. So if I summarize, these are the vaccines which are globally, the licensed and being used in cattle. So the two important criteria, this vaccine has as compared to the other licensed vaccine is that this vaccine is DIVA compatible, and it doesn't produce the side effect, which is being, all the case in the other vaccines as well. So it's highly safe, highly efficacious. It is a very good molecular signatures to differentiate it from the vaccine and field strain, as well as serological differentiation of the infected and vaccinated animals. It grows at a titer of more than 10 to the power 8, so economize the cost of production and provide immunity at least for over a period of 1 year, what we have seen. And it has no effect on the production parameters and can be used in the pregnant animals as well. So this is my last slide. So beside the vaccine development, we were also looking at the antivirals. So what we did is we propagated the pox virus for a long time in presence of an inhibitor, that is methyltransferase. So after 50 passage, so initially there was 3 log difference. So it was antiviral. So I was looking at a drug resistance. So after 50 passage, what happened, that the virus became resistant, completely resistant to the methyltransferase inhibitor. So that was actually disappointing in terms of developing is antiviral. But what we observed that P50 virus that is become resistant is growing at a 1 log higher titer than the normal wild type virus, or that was grown in absence of the inhibitor. So that was actually surprising, So that the resistant virus is growing much higher titer as compared to the control virus. So with a series of experiments, this is not part of this. So we investigated that the mutation was in the methyltransferase. So initially, we believe that this is the inhibitor is targeting the cellular protein, that is methyltransferase, encoded by cells. But later on, when we whole genome sequenced the virus, we observed that at that time we were not aware that this virus also encodes its own methyltransferase. And there was single point mutation in methyltransferase. So the single point mutation level is to grow at a higher titer. Why? Because that methyltransferase is responsible for the capping reaction at the 5' end. So the capping makes the viral mRNA more stable. So one of the problem what the Moderna vaccine was that, so to stabilize the mRNA, they are using pseudouridine. So if you are using the mRNA in higher quantities, so that leads to toxic effect. So one of the side effect that was due to because of the modification. Because mRNA is not stable, you have to make it stable, you have to modify it. So this is actually an alternate, and it's going to be game changer to stabilize the mRNA vaccines. This is something we already patented, and this is under also consideration in a very good journal for publication. Thank you very much.
Very good. Dr. Naveen Kumar compliments. Even though I am not a microbiologist, nor I have much expertise in your area, but as a public health person, and more than a public health person, as a farmer, I am really grateful to you for one, the efforts that have gone in to save the lives of not the cattle, but the families. I know the dependence of our farmers and their families. If a poor farmer has one cattle, one cow, and through the milk his family is being supported, if that cattle or the cow dies, what happens? I'm myself from the village, so I understand. And from that perspective, this effort has gone in such a fantastic way that today it is no more being discussed even in the media. In Maharashtra, there were situations and times where people were criticizing the government that nothing is being done. But the agriculture department of the government of Maharashtra gave these vaccines totally free, and today it is no more a problem. So compliments to the teamwork initiated, or the team which was led by you, NIV. So that is the, genuinely I feel this was a great effort. Now see, this entire session is designed in such a way that there are 4 topics which will be presented, and they are totally different from each other. So if you feel that it will be worth to ask questions related to the topic that has been presented, please go ahead, or else what we can do at the end of the session, we could also discuss or ask questions, queries, if you have any. But right now, if there are any burning issues, please do ask Dr. Naveen Kumar. I have a very serious comments, not on your topic, but on One Health. And that I will, that's the reason I didn't initiate the discussion. And I want to reserve and keep that at the end of the session, about One Health as a concept. And there are many practical issues that are coming up as far as implementation part is concerned, that I would like to take it up at the end of the session. So are there any questions? If yes, please go ahead, sir.
Question. First question is, how did you decide the titer of virus to administer in any cattle? And second, what's the storage condition? How do you stabilize your virus for long-term stability and transportation?
Actually, this is standard practice. This is even what is being commonly used for preserving the virus. So usually the stocks are preserved at -80. But for vaccine, because you need a supply chain, so every place you can't have a -80. So what we did is we freeze dried. So freeze dried the vaccine, you can put at 4 degrees. We have seen that at least for 2 years is stable without losing any titer.
And you don't need any adjuvant or any stabilizer?
No, this is live attenuated vaccine. You do not need any adjuvant. So it's clean.
How did you decide the titer value for?
Titer is being used because it's produced the cytopathic effect. So you can titrate, both TCID 50, tissue culture infective dose 50.
So dose is dependent upon the weight of the cattle, or like?
No. So, it's the vaccine, dose is not very much dependent on the body weight. So, the dose is 10 to the power 3.5 TCID 50, which is being recommended by World Organization of Animal Health. Although because this is a live attenuated vaccine, even if you use 10 to the power 2, so it works because virus somehow replicates, It's some sort of virus, but it doesn't produce disease. So the dose what we use is 10 to the power 3.5 TCID 50.
How long it stays in the body after administration?
Huh?
How long, once you administer to any cattle, how long does it stay in the body?
Virus?
Yeah.
Yeah, so as I shown in my slides, so we tried to, one of the key feature of a live attenuated vaccine is that the virus must not be excreted from the body. So that one of the limitation of the polio vaccine, you know. So polio virus is being excreted, you know, through fishes. So we have extensively examined that the virus was not found to be excreted by fecal, oral, nasal, any route. Also, you know, we couldn't see a significant viremia. In few of the animals, we could see that there is, if you do real-time PCR, 30, 40 CT values are there, but no, so it's fine.
Thank you.
There is no public health concern. It's also not being excreted in milk and even semen, you know. We also examine the semen of the vaccinated animal. They are all perfect, no traces of the vaccine virus.
Thank you. Thank you, Dr. Naveen Kumar. Okay. The second session is going to be, the presentation is going to be on topic role of genomic surveillance in novel virus discovery and linkage with One Health. This will be presented by Dr. Pragya Yadav. I will just very quickly read your biodata by the time you go there. So Dr. Pragya Yadav is a scientist F and group leader of ICMR NIV. She holds MSc in the biochemistry and PhD in biotechnology and has joined ICMR NIV in 2004. A field of research, virology, then molecular biology, public health, research and development, biocontainment science, molecular and serological assay development, new pathogen discoveries. Big list, huh? Okay. And lot of awards have been won by her. Rashtriya Vigyan Puraskar, Vigyan Yuva Shanti Swarup Bhatnagar Award, then Bhargav Vedanta Award, Dr. Vinod Kumar Bhargav Award in 2021, ICMR's Major General Sahib Singh Sokhe Award in 2017, and ICMR's Dr. T. Ramchandra Rao Award. Quite many awards, congratulations, and now you are going to make presentation. Thank you.
So thank you very much. I would like to thank Dr. Karaikal and Microcon for inviting us. And as I can see that I am very small in height, so I need to shift a bit. Okay, I think this is better now. So, so genomic surveillance using the One Health approach is a very, very new topic which is being used post-pandemic. And I am talking on high-risk group of pathogens. So if we talk about the discovery of next-generation sequencing, which actually, if you look at the history, it started in 2000. And that was the first outbreak, if you remember, that was SARS coronavirus. When it happened, people realized the importance of sequencing. But as the 21st century rolled out, we could realize not only the new viral diseases emerge, but the importance of tracking the pathogens, mutations, evolutions, everything become very important. There were another aspect which we learned with the experience, that the diseases which are under the eradication program, for example, polio. So if you are under eradication, you need to monitor not only the evolution of the virus, because whenever we try to use any antiviral vaccine programs, and try to do, the good example is antimicrobial resistance. The virus have a tendency to mutate more, and that's why it's become very important. Another example which I bring to your focus is SARS coronavirus. Initially when SARS coronavirus, I still remember when we detected in India first cases, everybody said that the virus mutations rate will be very, very low. But soon we realized after 6 months, the virus was mutating so much that it has thousands of the variants because of the mutation in one single type. So if I give example of the initial virus, initial 3 cases which came to in India, there were 3 cases, and from the beginning, out of 3 cases, there were 2 different variants with the mutation. And we had a lot of fighting between the groups of NIVs, because they were not agreeing our data to accept that the mutation happened itself in China. But after a few months, when more data came, genomic surveillance became very popular, everybody realized that virus was mutated in the beginning in the China. When the people leave, the virus left with them, and there was a many mutation which happened across. That was not all. If we look at the history of the mutations, anyway, I can talk, but you have to find the presentation for rest of the speakers. So if we talk about the genomic surveillance, and so if I just quickly describe in India, we have seen in 21st century many diseases, and if I just quickly summarize, you just remember after the SARS coronavirus one, then in 2009 we have H1N1 influenza outbreak, and that was the time when WHO declared that we should monitor influenza strains, understand which kind of strain is circulating, and based on those inputs and virus isolation, every year kind of vaccine would be taken for influenza is decided. So this become very, very important. Another example which I took it forward, in 2014, the Zika outbreak started in Brazil, where microcephaly cases started. And then there was a lot of challenge, is it the new strain or it is the old strain? And there was a lot of challenge. And then when people started sequencing, so sequencing data become very useful to understand if it is a new strain, because the microcephaly was not reported in the past, even where Zika was reported. If I talk about India, the Zika cases were when in 2017, when we enhanced our surveillance, the first case was reported from ANC case in Gujarat, and when we sequenced, we found this is not the strain which is circulating in other countries, but it was a very old strain matching with the Malaysia, which proved the hypothesis that Zika was present in India for ages. Only because of the lack of surveillance, we didn't know that which strain is circulating. But slowly, as soon as we could detect the outbreak in Jaipur, Rajasthan, Uttar Pradesh, we could see that the international sequences which was not reported in India was also present. If you look at the history of different viruses, so we continuously, we increase the surveillance, we can discover new diseases more and more. The most importantly in COVID, we also realize that it is not only important for surveillance, but this surveillance is important for your antiviral programs, your vaccine understanding. And in COVID, we all learned that how, when the variant of concern arrived, either is alpha, beta, delta, how everybody asked the question is the vaccine is working, the kind of vaccine we have taken, is the new COVID variant is going to work. So these understanding actually came with the time and people realized. There was another thing which I would like to mention, that in COVID we learned that whatever lockdown or efforts you make, the virus is going to move from one country to another because of the not only the kind of structure we have, but the kind of travel arrangements we have, the movements of not only the human but animals, vectors. And in this process, any variant which arises in any part of the world, we could see that transportation from one country to another country, within 2 months it was present in each part of the globe. So the virus cannot stop, pathogen cannot stop, and the only ways we have to monitor them. There are different kind of concept came in pandemic. You can do not only human surveillance is one of the way, but human, environmental and animal surveillance. And together, if we can screen them, it becomes one health concept for pathogen discovery. But at the same time, we also realize that pandemic, there is a new buzzword which come after this COVID pandemic preparedness. And pandemic preparedness become very, very important buzzword for everybody. And there is a lot of efforts which was not only taken by government of India, but across the globe. And people made new rules, new understanding, and which actually lead to understand more and more. So finally, they have got the presentation. Thank you so much. So I quickly maybe can take it forward. So this is because we are in Pune and I work in Pune National Institute of Virology. This is a BSL-4 facility in Pune. If you have get chance to sometime visit, or if you have watched the movie Vaccine War, you could have seen that. So we participate in many national and international surveillance programs. And the latest one which is started is called International Pathogen Surveillance Network, which is again based on genomic surveillance, and many countries are engaged, and NIV is part of that. The WHO COVID-Net, that is another surveillance program where we are monitoring the COVID strain to understand that which should be the next COVID vaccine. As a question, why we need a COVID vaccine? But for a understanding about the virus, still we have to keep tracking, observing, and the impact on people. If we talk about the NGS surveillance program, so it has a lot of benefit, which include not only the clinical care, infection control, public health surveillance, monitoring pathogen evolutions, and also vaccine preventable disease surveillance, AMR, foodborne pathogens, and also harmonizations with open species repository and analysis platforms. So I was mentioning about different disease emergence and how the genome sequencing become very, very useful in different outbreaks, and it started in 2000 and continuously it keep moving with more and more pathogens emerge, and we could understand more and more, and few of the examples are here in front of you. If we talk about genomic sequencing in crisis in India, so I would like to take you to the journey of COVID. When the first cases arrived in India, but prior to that also people asked, is India doing sufficient testing? Are we doing right testing? Why we are not getting cases in the January 30th when we did not declare the cases? So before even our first positive case report, we started sequencing all the people who came with the respiratory SARI symptoms from other countries, and we kept sequencing till we have, not only till we have the positive cases, but later on also till other laboratory did not pick up the COVID sequencing. And as soon as the first cases were reported, within 24 hours we could have the sequencing data and we could report to it. In Feb, further when we have more cases, and in March when we have importations from the cases of the Italy tourists in India, we could quickly not only sequence but we could inform. If you remember that our people were stranded in Iran, and we imported samples from Iran to India and we tested them, and then that's how the Udan mission was conducted, where people was brought back to India. They were evacuated, and those sequencing data help us to understand that what was happening to the genome of COVID virus. More importantly, as soon as we realized that COVID is a reality and it's going to stay with us, we wanted to have an indigenous vaccine. But to make a vaccine, you really need to characterize your virus very well. You have to develop all the indigenous tests, which was not available at that time in the March I'm talking. So we developed many tests indigenously within the NIV, and we characterize virus fully to understand how we can take up for the vaccine purpose. We have a collaboration with Bharat Biotech very quickly, and virus was detailed studies isolated from the Italy tourist, but it was from an Indian human being, and we characterize very well this virus and hand over to the Bharat Biotech for making vaccine. COVID also taught that how we can utilize the data from clinical to wet lab, and then how we can analyze and it can utilize for treating the patients or taking care how much impact it will make to them. This was the time when everybody learned about GSID genome sequencing data. People learned about what is the next generation sequencing, what is a different platform, and you know that nanopore, Illumina, and so on. So there are many sequencing platforms which everybody now can, even if you ask to the children, they will tell you what are the sequencing platform. There is, if you look at the kind of summation of data which people have done from 2000 to 2022, you can see how this peak is getting higher and higher. So we have more and more data. But we did not stop with the COVID. Now we have data for monkeypox, we have data for influenza, and all the new diseases, high-risk group of pathogen are getting added to that database. And if you look at the data, India was also participating fully through the GSET genome base in the and we established our own INSACOG genomic sequencing network utilizing different sources. So there were so many kind of consortia which was formed, and Asia PGI was also one of the popular one which was established in CR member state, and it was also in African country. But then WHO also came up with the guideline, very proper guideline, that how we can enhance the genomic surveillance across the different countries. And now, in the past, there were only few countries, especially developed countries, were doing genomic sequencing. But now, at least half of the globe countries, half of the populations are doing the genomic surveillance. And we have learned that genomic surveillance can help in the rapid detections, monitoring of pathogen transmissions, and also diagnosis, vaccine, and therapeutic by this. There is a data presented that which countries are working on which surveillance, and many countries are already working on tuberculosis and many vector-borne diseases, malaria, and those kind of transmission. And polio is also one of the very important disease which everybody is following where this program exists. If we talk about the COVID, after the COVID, when the variant of concern emerged, there was, as soon as the alpha variant emerged, there was more appeal to have the more genomic surveillance data. And India, we all know that in the beginning, we didn't have even sufficient labs to do the testing. And thanks to ICMR, Dr. Nivedita, who is a program officer for VRDL laboratory, they pushed this agenda at very, very high level, and we have now 165 laboratory. But at the same time, we also have 52 NGS laboratory across the ICMRs, then we have across the DBT, we have, so different ministries, they actually push this agenda of having more and more NGS sites, trained manpower, resources, and that actually help us to generate more data on understanding the COVID. That was not all. We also need to understand that when we are sequencing, how we are going to use this data for understanding, reducing the vaccine hesitancy, understanding the impact of virus and vaccine in those people who got 2 doses of vaccine, or if they got additional dose of vaccine, what is happening to the children, what is happening to old population. So there were many questions during the pandemic which was answered only and only that we had this genomic surveillance in capacity in the country. We could also develop different assays, and they were, I can just keep talking on COVID. COVID is one of the subject where NIV was on forefront. We did a lot of work. There was multiple teams were engaged in this work. But some of quickly I mentioned, the testing was one of the work which was taken up by NIV, the supply of kits across the country. We made many indigenous tests. We validated thousands of kits across, and then we did antiviral testing. More than 100 of the compounds were tested. Sewage water testing was taken up. And then we work on vaccines, and genomic surveillance was one of the major steps. But in this process, we also came across the mucormycosis, reinfections, breakthrough, and what are happening to after the booster days, what is the effectiveness of vaccines, and these were all linked to the variant of concern. Everybody asks, if the Delta infection is there, can we get the mucormycosis? If Gamma infection is there, can we get? So these questions cannot be answered unless and until you have the data. More importantly, when the vaccine, Covaxin, went through the phase 3 trial, there was a question if it is effective against Delta variants or not. And those data also came up, and we could even present the data that the vaccine was effective against Delta variant, which was the most pathogenic and deadly strain of COVID so far was presented. Another challenge came that can you prove it that if with the emergence of new variant of concern, if it is effective in the people who got the vaccine, and not only for the Covaxin, but for other vaccines which was deployed in India that include Covishield and so on, we all know the names. And so we took this challenge and we constantly isolated the virus. We did the genomic characterization, and we kept doing neutralization to understand that how they are working. Quickly, India could also receive the UL from WHO in 21 November, and we could have this vaccine that will become 8th vaccine in the world, which was approved by WHO. We could supply under the vaccine factory large number of vaccine doses with all these data that these vaccines were working against different variant of concerns. There was another episode which I would like to quickly emphasize when we started rolling out the vaccine to healthcare workers. Quickly there was a lot of infection happened to healthcare worker, and people started saying why we should take vaccine, we are all getting infected. So that was a very big challenge because this will create a lot of vaccine hesitancy. So we planned a study, and we took from the 17 state, samples were collected from 1 dose vaccinated individual, 2 dose vaccinated individual, and those did not take the vaccine but got COVID infected. And that those data actually help us to analyze not only the clinical symptoms, but also to understand what kind of variants were circulating in India. So if you quickly see this map, that was the time when mostly alpha variant was circulating, and some of the places we could see also emerging new or Delta variant. But this data actually proved that those people who got 2 doses of vaccine, they were not having severe disease, hospitalization, and that actually answered a lot of questions. Then there was another announcement made by many experts that the next, there will be a lot of deaths in the pandemic, in the pediatric population, because virus is going to amplify more, it will be more pathogenic. and then we did similar kind of study in pediatric population across the country. We could see that similar kind of viremia was there, similar kind of circulation of the viral genome were there. And that actually helped us to understand that there will be no different situation or many severe waves is coming in the pediatric population. We continuously monitor across the Maharashtra, across the country, keep doing the sequencing, and that helped to understand the circulation of different variant of concern and what are the strain which are causing more infection, which we realized when the Omicron emerged in 2021, and that also answered those questions. So constant effort it took to understand about the COVID pandemic. So now if we talk about, take a step back and think that was this kind of attitude we had in the past or not? So many times you think, no, we were not doing that. But I would like to change that thinking by presenting some more data, further non-COVID data. So in actually most of the country, not only India, we always work in the reactive. There is an outbreak and we run behind it. So these questions came post-pandemic and a lot of discussion, should we prepare better? Should we prepare more? So that we should not be reactive, but we should be prepared in a way that as soon as the case arrives, we should be able to not only detect, but we also able to respond in a very smooth manner rather in emergency mode every time. And the zoonotic diseases, everybody understand more now because of emergence of so many names which I mentioned, Ebola, SARS, COVID, they are all part of the zoonotic kind of emergence. And not only that, we could also look at the reverse zoonosis many times, and there are many examples of it. And WHO, even WHO, which thought only few diseases are priority and we need to prepare for disease X, soon realized we cannot predict like that. We cannot get biased. And now there was 300 experts deputed from different part of the world, and I was part of Bunia virus family, where they decided that which family and which member should be taken, which not only should be taken for prioritizing for diagnosis, but also for vaccine, antivirals, and so on. And these lists are there, and there is already work started, and many family-specific meetings are now going in different part of the world. And in Geneva next month, there is a meeting for each particular family, how we can take it priority, how we can fund it, and how we can prepare more for the future. One Health Joint Action Plan came with the many international agencies. They came together, and there was these 6 action track which was proposed, and it was decided to be achieved. And if you look at these 6 action track, the 3 tracks itself justify the need of not only the surveillance, but also how we can enhance our preparedness toward a pandemic preparedness. So if we look at the One Health concept, where we are considering everything, and actually being Indian, we all believe in Sarve Bhavantu Suvina, Sarve Santu Nunamaya. So, and I think I simplify One Health in the same format, that we are looking for a world where everyone is happy and we are not harming by any of the action to each other with our greed or with our development process. And in this process, if we want to have the surveillance, we want to have laboratory and policies, quality assurance, technical experts together in a way that can help to formulate the national strategy where we can do the harmonized sample and data collections, genetic material extractions, and harmonized SOPs, which the data should reach to the public health expertise and the proper action could be taken. We also need to have the national planning for pathogen genome surveillance, and that include a large number of things, but in summary, I can say that surveillance and laboratory system capacity, pathogen-specific characterizations, and more important, how we can utilize all the data in a form of One Health manner. Integrating genomic, clinical, and epidemiological data for enhanced surveillance would be one of the major steps which will be actually helping the pathogen preparedness for the future. So if you think that we have not done this exercise so quickly, I would like to take you for some of the success story of India with the COVID. And one of the stories is monkeypox, which arrived in 22 when we were dealing with the COVID very severe waves of Delta and others. We have now established 35 laboratories across the country. We supported CR member state for having a monkeypox laboratory. In-house indigenous tests were made, they were validated, and with the help of this network, which we could not only test, we could detect the monkeypox cases, but we could also do the differential diagnosis of varicella zoster virus, enteroviruses, and buffalo pox viruses. And this and the genomic surveillance could help us to understand that which strain is circulating in India. That not only include the strain which we detected in 22 and 23, but the recent strain which was further declared as a public health emergency, clad 1B. We also could establish the validation protocols and validate many industrial companies who are ready to make the kits for testing, and 3 of them are already approved by CDSO for making these products. We could establish ELISA, and which helped to understand the kinetics of disease, and this is the success story of very quickly detecting the clad 1B case, which was imported case, and this was India become 3rd country to report such cases, and the case is recovered, and there was no actually bad outcome. We could also start how we can establish environmental surveillance for the COVID, and the examples was presented by doing a hospital-based study in Delhi, and which could demonstrate that which are the area where monkeypox will be more, and what kind of precaution we should take when we are handling such cases. Another example which I would like to take quickly for Nipah virus disease. Nipah is a very deadly disease, and it has a very high mortality. India has reported many outbreaks that started in 2001 in Siliguri, then in 2007 in Nadia, West Bengal, and then from 2018 we have many outbreaks or sporadic cases of Nipah. Many times it is a deadly outcome, but our preparedness and surveillance has helped us in now detecting from the first index case, and that really helped to not only contain the disease, but also to stop the transmission and saving the life. But these efforts was not fruitful and useful till we have this approach of using all the C's, communications, coordination, and collaborations. And actually we followed in Nipah, and that coordination together become very, very important learning. And now we know more than what we learn in Nipah or other diseases in very long time, in very short time we could achieve in 5 years in Nipah, where we have clinical protocols for monoclonals. We are working with the industry and academia for making own Nipah, but we also know that where Nipah surveillance is required, what are the hotspots in the country by doing the BAT survey. We also know what kind of genomic sequences are present in which part of the country. So these information really help if you are making further any, if you have any therapeutics you want to do any trial, and all the stakeholders you can quickly map and they can be board very quickly. Though we don't have any vaccine so far available to use it, but we have different approaches and strategy interventions for Nipah disease exist in our country. Another disease which is not of human but of very important for animals, in 2018, just prior to pandemic, many gill lions died. So if a country which is 1/5 of the world population, we have only few hundred lions, only 500 lions. And our former DG said the lion's life is more important than us because there are few in the country. And 80 lions almost died. And we came to know that every year they die, and nobody knows why they're dying. So we requested them, then when this outbreak happened, please send to us those. They came to us, we did testing and sequencing, and it was discovered to be canine distemper virus, which is very close to the Asian genotype. And we could report back, and there was a lot of preventive measures were taken later on to save their lives. So these small efforts also can make a lot of difference for saving not only the human but also the animal lives. And this is another example which I would like to present. The next deadly disease, Crimean Congo hemorrhagic fever, which was initially detected in Gujarat, but we all know with the constant surveillance, genomic data, and a lot of data which produced in a few years, we detected first time in 2011. Many outbreaks has happened, but now there is a very set protocol, and we can easily detect from the index case, and the disease doesn't transmit to the rest of the population, and that really helped saving a lot of life. Not only that, we could also detect the imported cases in country, either in Kerala or in other part of the world. So this is India's preparedness for detections, and we could also know that what kind of resortment is there in not only in the human but also in the tick population, what is the seroprevalence in the animals across the country. We could study because of the genomic surveillance, isolation of virus we develop our own indigenous kit which is commercialized and can utilize for the testing of the virus. The next, we are sitting in Maharashtra, and Casanol forest disease, one of the zoonotic disease where monkeys, human, very good example of One Health approach. Every year, not only monkeys die, but also a large number of people get infected and they suffer. We sequence from 1957 so far, and we know now that this is a very conserved virus, but it still have some mutation of 3.5% of nucleotide variations. And if we make any vaccine because of very conserved genome, it should be able to adopt to it. In past, we have a vaccine, but now we are working on a new vaccine, and hopefully that vaccine will be available soon. We could develop different assays, which was transferred to industry, and it is available. During the pandemic, when we were dealing with the Delta wave, there was outbreak happened, and there was a child died in Haryana, and that's what India's first avian influenza case, H5N1 deadly case. This year, because of the very good surveillance, which was established by Dr. Nivedita's team, along with the NIV respiratory surveillance, we have found more avian influenza human cases in West Bengal, and this surveillance is going to answer more questions than we have in the past. So these are some of the examples. We all know that US reported this year that through the cattle, how avian influenza can transmit it to human, and that lead to a lot of worry about should we drink the milk or not. So surveillance answers a lot of questions related to the public health, and we should be definitely engaging in that. But not only that, surveillance can also answer many unanswered questions on undiagnosed death cases or clustering, or when things are happening like that. And I would like to refer to this clustering of acute encephalitis cases in Maharashtra, where we found 3 of the siblings died, and once we did the genomic sequencing and tested, we found that they were suffering with the rubella. And that was a very atypical presentation, and such kind of episodes were not reported in India. We also focus on many uncharacterized vector-borne virus isolates, and we could discover the new reoviruses we could isolate from mosquitoes, similarly from the ticks, and so on. So this list is very long. I can talk hours and hours on the genomic surveillance, but I'm just giving, Serum Institute has a huge horse stud, and there was an outbreak, influenza outbreak happened where there many horses died. Horses cost is in millions, and they are very, very costly. Those samples were referred to us, and when we sequenced, we found that is why equine encephalosis virus, which was not reported in India in the past. This again spread by vectors, and can lead to the death of horses. So far, we have also worked on bat surveillance in detail, and we found many viral pathogens which may not cause any disease today, but we don't know tomorrow. So those surveillance will answer a lot of questions. And I was mentioning about the Zika. Zika, many outbreaks has happened. In Pune has suffered this year. There was 100 of the cases which was recorded, and the genomic sequencing answered that where are the Zika present. Now if anybody asks that, is Zika present in my state? I said Zika is present wherever the mosquito is present, because we cannot, if we don't stop the mosquito, we cannot stop any viral transmission. And when we did this surveillance, we could also look at the co-infection of Zika, chikungunya, dengue in some of the cases. So many times when the people have the severe diseases, we don't know if they are suffering with not only one pathogen, but multiple infections, and that leading to very, very severe. So approaches for early detections have been taken by government very seriously. Now we have many coming up PM ABVIMS new facilities, and that one of the facility which I would like to mention, which Dr. Nivedita will take lead and describe more, that there is a National Institute of One Health coming in Nagpur under the One Health Mission activity. This is monitored by PSA office, and ICMR is coordinating this, where 13 ministries across the country would work together, and also all the state governments to have a prepared country better for pandemic, improving our surveillance program. And I'm sure this is a very, you may think that this is a very bright picture which I am showing, but I want to see this picture to be true, and I want to see this happen in our country. We have a better surveillance, and we should be able to save a lot of life, which we are losing every day because we are not able to detect, we don't know about them. And I'm sure these efforts will take us to that step. Thank you very much for the opportunity.
I can tell you, being part of the DHS for many, many years and NIV as one of our family contacts, you can say, and we depended so much on NIV ICMR. The work that has gone in, particularly in the last few years after COVID, is great. And I feel the direct benefit to the community that what you have presented are quite reflective that yes, things are moving. However, there are a lot of challenges. When you last slide that you presented with 13 ministries, in our case, you know, when we used to say that coordination is very important. You have to collaborate. The first question is you coordinate with me. In the medical college, one department to another department, the coordination responsibility with the other department, not with me. With that concept that we work in India to have 13 ministries and X number of people within those ministries, God help us. I'm a very optimistic person, but I know I've spent 40 years in the administration, so I know what the situation is. But however, the progress that we have made post-COVID is very, very wonderful. And the only thing that we have to remind ourselves is that, are we really serious about this? You know, once upon a time, people who did not know what do you mean by, even don't know the spelling of genomic surveillance, and we used to conduct the meeting, the great politicians. Yes, I've worked with these guys for many years. And that time, this buzzword, you know, rapid test, surveillance, isolation, vaccination, everything was being discussed from top to bottom and appreciated by everyone. Now everybody has forgotten to the great extent. That's the ground reality. With this, are there any specific questions for her? Because the way she has presented is quite comprehensive. But if there are any questions, clarification that you want to ask? Right?
So I bored them and I've...
No, you did a great job. Thank you. So there will be now third presentation. Friends, we are slightly getting delayed. Are you okay with that?
She completed the advanced vaccinology course in France and epidemiological evaluation of vaccines in London. Also, she is the certified auditor for the implementation of the WHO Containment Certification Scheme for Global Action Plan 3 for polio. And she holds many professional memberships, and she received awards both Indian and international from Indian and international societies. And she has been frequently represented ICMR internationally. And she has key contributions in various missions. The list are very extensive. I would like to enlist few like COVID National One Health Mission, COVID-19 pandemic, especially COVID-19 testing and COVID-19 vaccine development and detection of Zika virus in India, detection of Nipah virus disease in India, and upholding polio-free status in India. And she is having many scientific publications, around 158 scientific publications for her credit. We welcome you, madam, to talk on role of laboratory in integrated One Health approach for public health.
Yeah, thank you very much for the kind introduction. And this font also of my slides has changed, but I'll just go ahead. And I would be presenting the role of laboratories in integrated One Health approach for public health. Now, if you talk about the role of laboratories in public health, I look at it as a multi-pronged and multifaceted role. Definitely, laboratories do have a role in detecting the etiologies in various samples which are referred to them from patients and which aids in the appropriate management of patients. But besides that, I feel that a much larger role that the laboratories in today's context play is the determining the etiology of various outbreaks that happen all across the country. And I think a very critical role of laboratories, which I don't think is very well understood till date, but I think the role of laboratories in testing samples of outbreaks for prevention and control and timely instituting these measures is extremely important. And laboratory is also the backbone of all the surveillance programs. not only to depict the trends of various infections that happen all over the year, but also laboratories are very critical in understanding and quickly detecting the new or novel pathogens that keep on emerging all the time. So I think it has an enormous role. But the prerequisites that each laboratory should have, and particularly with reference when we talk about the One Health context, I think it's very important to have very strong linkages with relevant public health programs, state governments, so that you get samples and samples with proper clinical details. Unless a laboratory has access to clinical details, those samples really don't mean much. And then obviously it is very important to keep pace with newer technologies which are emerging every day. We just heard novel, the genome sequencing, and then there are many, many other technologies. We heard about microRNA from Dr. Naveen. There are new molecular platforms and many more that are coming. Now, the major challenge that we face in India is, though we have thousands of laboratories all across the country, but when we go on a hunt for well-characterized samples for doing various studies and also validating our newer diagnostics, we never find biorepositories. So I think that culture of maintaining biorepositories as we venture into more and more newer technologies so that well-archived, well-characterized samples can be accessed, I think that's extremely important. And while we do all our work, we really have not such a great focus on maintaining quality, but I think from day one it is critical to have lab quality management systems and adhering to the quality control programs like internal lab quality controls, external quality assurance programs. Also have a very good understanding of biosafety and biosecurity. That's all messed up all across the country. There are few apex laboratories which follow it pretty well, but then downstream what happens in terms of biosafety and biosecurity, that's very concerning. So moving on, and when we especially talk about One Health approach, detecting novel pathogens and working across sectors, it is very important to move with a sound background of biosafety and biosecurity practices. And also along with this, it's very important to keep on retraining the staff. There is a lot of staff attrition in laboratories all over, and we often forget to train them and they learn on the job, and that's where the errors happen. And that's why it is important to have an internal audit system for lab operations and data quality check. As we just heard and about the skepticism around the One Health approach, it is important to work collaboratively. We should be open to exchange samples, exchange information, and not feel threatened all the time. I think working collaboratively is very, very important while embarking on a One Health approach. Be open and enthusiastic to newer ideas and having inquisitive mindsets. Now I'll talk about few very key national networks which I have been coordinating at ICMR, and they are an integral part of the National One Health program now. One is definitely the DHR ICMR Virus Research and Diagnostic Laboratory Network. Now after the 2009 pandemic, when the pandemic happened, there was only NIV Pune and to an extent NCDC Delhi which had the capacity to test for influenza viruses at that point in time. And the country of a size of India was really grappling to find out means and measures to actually test various individuals who were suffering from symptoms. And at that time, I think this was a wake-up call for the government of India, wherein they decided to roll out a structured scheme to set up viral research and diagnostic laboratories all across the country. We started it as a small program of ICMR in 2010, and it became a full-fledged scheme of Government of India through the Department of Health Research in 2013. Now there are 165 VRDLs all across the country now, and you can see the spread up of these labs in the map that is there on the right-hand side. And this is a 3-tiered structure wherein the regional level labs are the highest tier, followed by state level and medical college level. Now, these labs are involved in timely detection, diagnosis, and outbreak investigations, training and research of known and novel viruses. And also, they are very actively monitored. There is an APEX center at NIV Pune, which trains and retrains the staff of the VRDLs, give them SOPs, and also troubleshoots on a regular basis. And data mining is housed within the ICMR Institute at Chennai, which is the National Institute of Epidemiology. Now, this network has tremendous contributions starting from its inception, and in the interest of time, I did not make a separate slide, but it's huge in terms of especially the COVID-19 pandemic, when we had 106 labs when the pandemic started. And all these VRDLs actually went into massive trainings and capacity buildings and exercises at the level of states and regions, wherein they trained every possible lab, be it from the private sector, government sector, and that's how from a lab, one lab at NIV Pune and 30 labs in February 2020, and we could reach a number of 3,300 molecules of DHR ICMR, which are either having a BSL-3 or are in the process of establishing one. And we also ventured into setting up a mobile BSL-3 lab. This was a very active collaboration between CleanZeds and CleanZeds Contamination Control and ICMR, wherein we could actually make the Asia's first mobile BSL-3 testing lab. And I will show you the interiors of this lab, and Dr. Pragya's team has immensely worked with it. But it's the state-of-the-art facility which can go in any part of the country where the outbreak is actually happening. It can go even to the forest fringes and has all the capacity to test samples wherever the outbreak happens. So with this laboratory capacity, it was deployed twice in the Nipah outbreaks of Kerala, and it tremendously reduced the turnaround time for testing, wherein every sample was earlier referred to NIV Pune, took 72 hours to diagnose, but with this asset, we could actually diagnose it within 6 to 8 hours. And we have 52 NGS facilities all across the country now, which are being used under the One Health program, and these capacities we are trying to utilize. And one of the greatest things I would say, and I am very fortunate to be coordinating this initiative from the ICMR side, where we went into scaling up and setting up a lab in every district of India. And we had a huge program along with 14 mentor institutes in institutes of national eminence all across the country, wherein we succeeded practically in establishing labs in almost 90% of the districts all across India. And realizing the importance of molecular testing labs, the government, in fact, the highest leadership of the country took stock of the situation of molecular virology labs in India, and they were actually appalled to know that very few of the medical colleges at that time had a molecular testing lab. So this Gazette notification came out in June 2020, wherein the government of India notified that for like certification or accreditation of any medical college in India, it would be mandatory to have a molecular testing lab. So I think this was one of the biggest victories in terms of making the leaderships understand the importance of having a molecular lab. And this is what phenomenal escalation in the laboratory capacity we saw during the COVID-19 pandemic, starting from one lab at NIV in January 2020, and within a span of 2 years, we reached a figure of 3,500. And they could also, besides testing, they tremendously contributed in validating various newer diagnostic tests that were being made by startups and companies in India. And because actually ICMR had set up 30 validation centers all across the country, they were validating all kinds of diagnostic commodities, be them molecular tests, rapid antigen tests, ELISAs, as well as even the swabs and BTMs which were being made in India. And because of this phenomenal movement that happened with everybody coming together, the prices of the RT-PCR testing, which were 4,500 in early part of the pandemic, really came down to rupees 200 within a span of again 2 years. And the rapid antigen test, when we first approved at ICMR, it was 450 rupees, which dropped to 10 rupees. So this was a massive reduction in prices, which was actually due to the efforts of many people sitting here and those not here as well, who actually validated these diagnostics day and night. And many people, many government departments funded various startups and various industry to make these diagnostics for use in India. And during the COVID-19 pandemic, the environmental surveillance also actually got a new dimension. This was only being done for polio earlier than SARS-CoV-2 pandemic, but during the pandemic, this was massively done by many departments. Even the INSA-CoG Genomic Surveillance Consortium picked it up, and many people were doing this sequencing. And moving on, in the National One Health Mission, there is definitely a very, very strong placeholder for environmental surveillance of not only SARS-CoV-2, but all the emerging, re-emerging infections, and also novel approaches for understanding that how can we discover unknown or novel pathogens from wastewater are also being explored. And as Dr. Pragya briefly touched that after the COVID-19 pandemic, there was a very structured thinking within the Prime Minister's office and senior leaderships that with this kind of a reactive approach, a country like India cannot move ahead. It is very important to have very strong surveillance mechanisms and mechanisms to pick up any outbreak in very early stages. And that can only happen if we embark on an integrated One Health approach. And this was recommended by the Prime Minister's Science and Technology Innovation Advisory Council. And very unanimously it was decided that the secretariat for this National One Health Mission would be housed within Department of Health Research and Indian Council of Medical Research. And this is already shown, which is actually talks about having 13 stakeholder departments together. And just Salunke sir, responding to your concern, I was also very skeptical when this came up, and so all of us were. But if we look at the enthusiasm when we talk to the states and when we talk to the people in animal husbandry or environment and forest who work at the level of states, I think the enthusiasm to collaborate is excessive, and unfortunately it was never tapped. So while this mission brings together a lot of collaboration at the top level, but we also work from a bottom to top approach, wherein we catch hold of all the relevant stakeholders in respective states and we work with them. And it has been an overwhelming response, let me tell you, which we also never expected. And this gives us a lot of hope that this mission will really see the light of the day. So situation is definitely not that grim that we used to think. Now, under this National Health Mission, there are several important objectives that are being undertaken, but I would restrict myself to the laboratory part because that's the topic of my talk today. So under this National One Health Mission, there is, it has been pledged to create a national network of biosafety level 3 laboratories. Now, with this national network means that every department will contribute their best laboratories to this national network, and we will have a One Health network under this initiative, wherein suppose if there is an animal husbandry laboratory in a particular location where an outbreak is happening, then the samples in the first place would be referred to the animal husbandry laboratory, and this animal husbandry laboratory would be competent enough to test for human samples also. It's not that they will test only for animal samples, and the necessary trainings, hand-holdings, all are happening within the mission to empower each of the laboratory to do all kinds of heterogeneous testing, and they becoming very pluripotent kind of a resource. And similarly, the human laboratories at ICMR, DBT, etc. are being trained for animal sample testing, so that any outbreak sample which is referred to them, they should be able to test. they should have appropriate reagents, kits, everything. For that, budget is also being allocated, and necessary trainings and hand-holding are happening for that. So the whole idea is that the outbreak should be diagnosed at its earliest. Wherever it is happening, send the sample to the nearest lab and get a very quick diagnosis so that the response to the outbreak is not delayed and the transport issues of samples are circumvented. So currently we have 22 laboratories under this network, wherein ICMR, DBT, CSIR, Department of Animal Husbandry, then Indian Council of Agricultural Research, all have contributed their laboratories to this network. And the first training to sensitize this laboratory network about biosafety, biosecurity practices of human sample testing was conducted in April 2024 at NIV Pune, where all the laboratories participated, and it was a very successful training, you can see, where all these 22 laboratories were rigorously trained over 2 batches of 1 week each, and lots of hands-on exercises and simulations were done. Then a similar training of all these laboratories happened at NISAD Bhopal, which is the high security animal disease testing laboratory at Bhopal. It's a BSL 3+ laboratory. So Bhopal undertook training of all these labs to test for animal and environmental samples. And now with all the priority pathogens enlisted on the human and the environment side, they are also being provided with the necessary reagents, kits, so that they will be able to test samples from different origins. And also, once these labs were trained, it was actually a training of trainers, wherein we expected each of the lab personals to go back and train their respective people in what they had learned. And in order to ensure that they are really doing what we actually have trained them to do, we have a tool now, and this tool actually has been developed to assess BSL-3 laboratories. I won't go into the details of this, but it covers each aspect of how your staff has been trained, how you receive samples, how you aliquot them, how you handle them till you test them, and eventually how you dispose and store them, and also give a report. So it has many subcomponents, but this comprehensive tool has been developed by us, both ICMR headquarter and NIV Pune together, and wherein we have now a very objective way of assessing the BSL-3 laboratories under the National One Health Mission. And then the other most important part is that when you are handling a lab, you should have the standard operating procedures to actually test. And so for that, we took various SOPs which were sitting in the animal labs, human labs, and also the environment testing labs, and we have made a compendium which is now put in a common domain, and it is accessible to all the partners of the National One Health Mission and those who are out of the mission as well. So this provides you a very comprehensive SOP repository for testing anything that you may want to test across sectors. And moving on, the whole approach is now towards syndromic surveillance, wherein we, in a One Health approach, we really don't want to embark on a surveillance which looks only for one pathogen. Here we want to look at the spectrum of pathogens which cause a similar kind of illness. And for this, we have enlisted these 7 syndromes which you can see on this slide. And for each of these syndromes, we have actually developed a list of priority pathogens and also a very comprehensive syndromic testing algorithms where we could actually test for everything that is known. And these are so comprehensive, these algorithms, that eventually they drive you down to a proportion of samples where you reach the unknown proportion. And that is where we hope to find a novel or a new pathogen. So it's a very comprehensive approach, syndromic surveillance approach, where we follow an algorithmic design to reach to a decision or conclusion of an unknown sample being present in our clinical specimen. And for this, the syndromic surveillance has been now rolled out by ICMR along with all the relevant stakeholders of the National One Health Mission in slaughterhouses, zoos, and bird sanctuaries. Slaughterhouse and bird sanctuary project we have already started, wherein the animal husbandry, the Ministry of Environment and Forest, and all the necessary stakeholders are there. And we are looking at spillovers across human-animal interface here, using our syndromic surveillance algorithm. And we strongly believe that this may be a very effective way to detect spillovers very early and document them, and also create a repertoire of pathogens which cross animal-human junctions. So all this has been started, and these all surveillance programs have been started with strong wastewater surveillance programs for whatever we know and whatever protocols we have, and we are still evolving to make these protocols. And there's now what is happening is that while we are expanding our syndromic surveillance program, we realize that the pathogens that we want to test, we don't have diagnostic tests for them in the country. So along with this, it is very important to scale up the capacity for production of diagnostics, in-house affordable diagnostics in India. I would probably skip this slide. And for this, many approaches are being used, which are nanotechnology, PCR-based approaches, CRISPR, whole genome sequencing, AI-based diagnostic approaches, sensor-based approaches, imaging-based approaches, then point-of-care, multiplex, portable devices. There are many. And Indian industry has become so vibrant after the COVID-19 pandemic that every day they would come to us that we have this solution, please validate it for us. So we are also within the One Health mission, we are trying to create capacities for validating these diagnostics, having biorepositories. And for biorepositories, what step DG ICMR has recently taken is that select number of VRDLs are now being upgraded into infectious disease diagnosis labs, wherein not only viral samples will be there, but bacterial, mycology, parasitology, all these verticals have been added to the VRDLs to make a very comprehensive framework of diagnostic testing. And they would also have structured biorepositories. They would also serve as a resource for validating diagnostics. All that has been given, they have been given a task. And I will just not talk much about it, but we are working with many diagnostic manufacturers across India to set up panels for multiplex testing and doing a very effective syndromic surveillance under the One Health mission. And under this initiative, we are also working with the Drug Controller General of India to establish 60 medical device testing labs across the country, along with biobanks, etc. So thank you very much for your patient listening, and thank you very much for the opportunity.
Thank you. Optimistic. I wish to join you in that optimism that this One Health will really work at the cutting edge level, state, district and below district, because that is the cutting edge level.
Yes, sir.
The problems really start from that level to build the capacity of the peripheral most functionary, both in the health sector as well as animal husbandry or animal, agriculture ministry or department, and sustain that. What happens, because many years have been spent, so slight skepticism is a part of that particular experience, that committees at the level of the chief secretary, district magistrate or the district collector is one position in this country which is most abused. That person is expected to chair 65 committees, and because he or she is expected to chair so many committees, the kind of attention that is being paid, let's not discuss that. The point that I'm trying to say, that this looks so fantastic and so wonderful that somebody said initially, Sarve Sukhina Santu, Sarve Santu Niramaya.
She said.
Yes, sir. So that is likely to happen, but it is not wishful thinking alone. It requires to have support of resources. With hardly 1% of our GDP, if we are going to spend in the public health, how do you expect that will reach? We should reach, but at the same time, why should we wait for another COVID? We had enough. I tell you, we are all very lucky. I have my own near relatives, I lost them. People from ICMR family, we have lost them.
Yeah.
People from WHO, we have lost them. DGHS, additional DG, good friend of mine, we have lost. We have lost a lot of people we can't afford to ignore the impact of COVID, which was so bad. But today I feel that it is our responsibility at the appropriate level to say that yes, our directions are good. One Health as a concept is excellent, but are we backing it with the kind of resources, capacity building and prioritization? Kindly allow me to make this particular statement. Our politicians, with due respect to them, great guys, but they are only derived when people start dying and media start, howling. That is the ground reality. How do we ensure that they sustain this interest from the longer perspective? That's the only limited point. You did a great job, as usual. We know that ICMR does really a great job. So friends, we are about to now finish this session after one more presentation. Do you have any questions to Nivedita? Somebody said no. That means she has done a great job. Thank you very much, Nivedita. She did a great job. So the last... Public health response, a topic of great interest to me at least. Dr. Mala Chhabra, she is Associate Dean in Delhi. Which institute, madam? Excellent. So she is, I can only indicate the great kind of work she has done with 37 years of research and teaching, training experience she has. She has a lot of publications. If I cut short your biodata, it's okay, na? So, lot of awards, a lot of research work, but we would like to listen to you. Yes.
Good evening, everyone, and I wish to thank you first that you are still here despite a very busy day. And I am the last speaker, but not the least one. I am very sure about it. So thank you so much for being here, and I really want to thank the organizers and Microcon D for inviting me here. Amongst the august speakers who have spoken, I will take you to the field. I am a microbiologist by profession, and I was additional director and head of zoonosis division, worked at NCDC for 18-19 years, and now I am at RML Hospital, New Delhi, and I am part of the VRDL. So I have both the cakes. I have seen what the public health microbiologists can do in the field and what are the challenges that we face. And on the other hand, now working with ICMR on VRDLs to come up with the surmount these challenges that we faced in the field and see what best we can do for the country. So with that, I think they need some help with my presentation.
I got it. One help.
Yes, please.
You should recruit somebody permanently till the session get over. This is not a good example.
And how should we respond to them? When we talk about public health emergencies, it clearly indicates that it could be a disaster, it could be an earthquake, it could be a flood, or it could be disasters due to microbes. So it could be any disasters, but today my focus would be on various microbes that have caused havoc in the country, and I would talk about the response in public health emergencies. And these were, some of the things were mentioned by the previous speakers. These were the public health, about 13 in the last 3 decades, starting 2000 to 2024. We had these, and if we carefully look at them, there are 13 of them in these, which means every 3 years you're getting something new, something challenging, something you need to tackle. And of these 7, the one mentioned in the red, are public health emergencies of international concern. Another very important point to note here, that most of them are zoonotic in nature. So there are 60% of the existing diseases are already zoonoses, and 75% of the emerging infections are zoonotic in nature. In addition to that, they have potential for bioterrorism. So as Dr. Nivedita said, biosafety and biosecurity becomes extremely important. And of these are the viruses which are most abundant, biological entity, and now most important cause of epidemics and pandemics. So what is the viral burden? Can you count the zeros? So it is about 10 to the power 31 viruses that are circulating in nature, and someday as I go in my presentation, will realize that all of these have to be taken care of if we don't take care of ourselves today and respect ourselves today. So important viral events in last 4 decades have been due to novel viruses. These have originated from wildlife, global, and they have impacted the global health and have had economic implications, and that is the bottom line. That there are so much of economic implication in that we cannot afford, as Dr. Salunke said, only 1% of GDP going in health budget. How can you afford these pandemics? And in last 4 decades, have been era of viruses. As I said, 18 of new viral human pathogens identified in last 40 years. 12 of these have originated from animals, and 11 of these new viruses assumed serious global importance, especially in the current millennium. Now, just picking a few examples, this was 2003, SARS came to, and this was the first coronavirus that spread. We already had 3 or 4 coronaviruses circulating, but this was the first one, and at that time, I used to put in my slide in bold that this was the fastest spreading virus. It affected about 8,000 individuals, 800 deaths, 26 countries in 8 months, spread through flights. People got infected. And of course, after that, SARS-CoV-2, brought down this impression, and we knew what happened. For SARS-CoV, we had about 3 probable cases, 10 suspect cases, and no confirmed case, and we were very, very lucky about it. Then was avian flu. Somebody has already touched it, and this H5N1 has, it has caused loss of about 10 billion USD to avian poultry industry in Southeast Asia region. And in our country, it started in 2006, and subsequently, the country, 2/3 of the country has reported outbreaks due to avian flu, and we've taken all the action. Department of Animal Husbandry has made the plan, contingency plan to deal with it. Why I'm bringing this up is this is the first time that intersectoral coordination was very effectively established between Department of Health and Department of Animal Husbandry. I started my journey at NCDC in 2000 in zoonosis division, so I was very keen on zoonotic diseases and wanted some of the answers from Department of Animal Husbandry. That time I used to call IVRI, and it was one-to-one, talk and guidance. Despite our best of attempts, ministries did not get involved. But this was, first turning point, and this was the highlight. Even today, the RRTs comprise of, along with veterinary people and the colors, one person, human health person, is there to monitor the health of the colors, which is very, very heartening, and one of the important things that avian influenza brought to the country. And now we have human cases of avian influenza, as Dr. Pragya already mentioned. What does this mean? This means that we have to strengthen surveillance to avoid human cases, because we've seen the examples of, what happens when human person gets avian influenza. It becomes a very serious issue. Now, one morning, we know when we had avian influenza in poultry, we culled the birds. One morning, I get this picture in my email, early morning when I opened the email, and said that so many crows, the sweeper who was sweeping said, "Madam, so many crows are dying here," because we went there for a training. So he told his supervisor that so many crows have died. So they clicked the photograph, gave it to their district surveillance officer. He sent it to us, "Ma'am, what to do?" First thing that came to our mind was West Nile fever, because in US we had seen that crow deaths preceded acute encephalitis syndrome there. But that time, as I said, nobody would take responsibility of crows. Department of Animal Husbandry said it is not our responsibility. Wildlife Institute said we don't work on crows. Where do we send the sample? Then the National Institute of Virology was contacted, and they tested, and found it to be the H5N1 in crows. Now see how the viruses behave. We were culling the poultry because we could hold them and do that. But what about the crows? How to deal with them and how to cull them? So viruses will also find new hosts, and we have to be careful and deal with them. People have, previous speakers have spoken about the influenza pandemic. H1N1 started in 2009, but we were all prepared to deal with this pandemic because of our previous experience with the avian influenza. Labs were prepared, ICMR strengthened the labs and the VRDL network, and one could deal with them. Then the very important one was this Hendra and Nipah virus, and this shows, you know, the places where Hendra and Nipah could occur. Let me take you to a bit of history. The virus was first discovered in 1999 when the outbreak occurred in Malaysia. I will bring in another component here. How did this outbreak start? And as the legend goes, that people were burning in Indonesia for urbanization, they were burning forests. And there was a lot of smog everywhere. So that year, the fruits, the trees did not have flowers and fruits. Entire bat population from Indonesia flew to Malaysia. And Malaysia, there were a lot of fruit orchards, so bat happily habitated that. And for land use, they had piggeries in the orchards. So these bats, under stress, excreted the Nipah virus, and the piggeries, all the piglets and pigs, they got infection, respiratory infection, and their respiratory symptoms, the cough could be heard up to 1 mile, and it was called 1 mile cough. That is how this outbreak started. And then, because of the import of meat, it went to Singapore. The outbreak occurred in Singapore also. But the kind of action that was taken, all the pigs and piglets were culled. And they did not have any other outbreak after that in Malaysia or Singapore. But the adjoining areas, the virus kept circulating, and Bangladesh has huge number of outbreaks of Nipah virus. This slide was also shown by Dr. Pragya. You know, the first one, Siliguri, 2001, we got the samples, and first thing that stuck to us while at NCDC was that this is plague. So we tested for plague, negative. We tested for leptospirosis, negative. We tested for scrub typhus, turned out to be negative, but it was highly infective at that time with a lot of mortality. And then we tested for some exotic viruses like Rift Valley fever, Hunter virus fever, because we had the reagents at that time. 2 years later, we realized that this was Nipah virus. Imagine, at that time, we tested them on open benches. So standard precaution becomes so important when we work in the labs. As Madam said about biosafety and biosecurity, that has to be taken care of if we have to work properly in the labs. Subsequent outbreak in May 2018 in Kerala, where there was 90% mortality, was again we were in field, and number of challenges faced were that we didn't know how to catch the bats because only one institute was doing it. So to transport everything there and then catch the bats, bring them back to the National Institute of Virology to test them, or to Nishad Bhopal, was a big, huge challenge at that time in the field while we were in the field. But NIV team did a fantastic job at that time. But what I want to say is at that point of time, initially there was a lot of challenges. The last outbreak, September 2023, again, I was in the field, and we've had, again, 6 cases, 2 deaths. Mortality was about 33%. And when we went to field, this was very interesting. The primary case suffered from Nipah virus disease. He had come back from Middle East countries, and he was a rich man, had fruit orchards, and the familial household cluster was his son as well as brother-in-law, who subsequently recovered from the disease. But the hospital cluster, now these outbreaks involved the hospital, the healthcare workers. There were 3 cases in hospital, one of them died, and that was a bystander. He had nothing to do with patient. He had just come to the hospital as an attendant to another patient. And 2 cases, one of the healthcare workers and another bystander, they recovered. Now to investigate as to how this spread was like a CBI investigation. We looked at all the cameras, CCTV cameras, to see that where he got in contact with the patient. And we found out that he was looking at the patient when the patient was being intubated. So he probably got the viruses there. Again, break in the standard practices. This thing was that how did this primary case get infected? So we went to the field. We found so many bats roosting there. But they are there for years. Why today this case has occurred? How has it occurred? Why the bats are secreting the viruses? We had wildlife experts, but we did not find the answers. And these are the answers that we have to look, so that, because we should remember that fruit bats are present everywhere in the entire country. So if we have to find out as to why these outbreaks occur when they occur, is a bigger challenge for us. That is why we should understand the habitat and the nature of the bat. As Dr. Pragya has mentioned, bats hold so many viruses. But, which one, when will it become a disease or zoonotic transmission, we do not know as yet. I will briefly mention about Ebola virus, which, did India have a case of Ebola virus? Anybody? No. Excellent. So, Ebola virus disease till 2013 was only confined to villages of Africa. But this was totally unprecedented, because this was the first time that it came out from villages to the city and then spread to many countries. And it was declared as public health emergency of international concern on 8th of August 2014 and lifted in 16. 2 years. And India was all prepared, and we were in the pre-epidemic phase. We were screening patients, and the characteristic of this was that exit screening was started during the Ebola virus. They realized that people should be screened while they are exiting the country instead of when they are entering any other country. So same thing was being done, and these performers were developed to screen these patients. And another good thing about Ebola virus is that they get transmitted only when the patient gets the symptoms, when the patient becomes symptomatic. So unlike COVID or other respiratory, when they get transmitted during the incubation period also, this was a big advantage. So we screened about 106 samples of the people who had some kind of symptoms when they traveled from these countries. And then, because there was an exit screening, they were carrying people who had suffered from Ebola virus and recovered. They carried a certificate that they were recovered case of Ebola. So we collected samples of sweat, of saliva, blood, and semen from this patient, and we tested it. And per chance, I was in the laboratory in the semen. When I saw the graph rising, my heart was sinking, because this was the first case of recovered Ebola case showing the virus in the semen. And the sample, because we had a very good BSL-4 facility in the country, in NIV Pune, the sample was sent there to see whether we are detecting the antigen or it is a live virus. Now, this was a young boy traveling from Western Africa, going to a village to get married. He was quarantined for 5 months, and every 15 days his semen sample was tested to see if we were detecting the antigen, and it was tested in 2 labs. We were sending the samples to NIV Pune also to see whether the virus is live or not, and that time WHO said that that you have abstinence from sex for 3 months should be done after a case recovers. But this case had the virus detected, genes detected till 5 months. And subsequently, as Dr. Pragya once told me, that WHO changed the guidelines because of this, and we together have, NIV and NCDC have co-authored this paper in EID. So then we continued to have, in 1920, there was another Ebola virus outbreak in DR Congo, second largest with 2,300 deaths. India was prepared, but we did not have any issues because the outbreak did not spill out of DRC. But now see what happens. Till now, it was Zaire strain of Ebola virus which are circulating. Now the strain changes. It is now the Sudan strain which is circulating. So viruses will have their way of changing and surviving. Then comes the Marburg virus. Again, this is equally deadly as Ebola. We do not know much about this virus, so we have to be careful. With this, I think two other viruses that we have to keep in mind in our country are Crimean Congo hemorrhagic fever virus, because they have not become of international concern, but they are definitely of our concern. Why? Because when we went to 2011, when the first outbreak occurred in Gujarat, NCDC team went to investigate that, and there was this lady who suffered from viral hemorrhagic fever. She was admitted in hospital, died. That's it. Subsequently, when the doctor, the staff nurse, and a person who met the staff nurse suffered and died due to disease, there was panic. And then it was diagnosed as Crimean Congo hemorrhagic fever in the healthcare workers. So healthcare workers get involved in this, and NIV, this is the NIV slide, they told us that they had already detected IgG anti-positivity in livestock in Gujarat. And now Gujarat reports outbreak every year and they manage the outbreak. The outbreak occurred in Rajasthan. I will again bring the environment here. How did the outbreak happen in Rajasthan? We were in field in Rajasthan, and when we took the history, we realized that the feed was transferred from Gujarat to Rajasthan, and this feed had infected ticks. And the outbreak occurred in a person who was distributing this animal feed to the farmers. In UP, outbreak occurred, we could not trace, but we heard that there was a lot of animal exchange in UP in the animals, and so there were a lot of ticks. We could not isolate the virus from the ticks, neither we could detect the virus in the tick, but we could sequence the virus from the lady who died of this thing. Now there are multiple outbreaks in Kerala. What does it mean? You know, subsequently there's a publication, we did the sequencing of this virus, and see, it is coming from our neighboring countries, Tajikistan, Uzbekistan, and there is free flow of animals in this. So there needs to be not only intra-country collaboration, there has to be inter-country coordination to see how we can stop this spread of zoonosis virus. And this was again published, and subsequently we've seen that the seropositivity of Crimean Congo hemorrhagic fever in livestock has been detected in 22 states. And not only in livestock, but also in sheep and goats. What does this mean? That the virus is circulating, it is only a matter of time, and we do not know what are those factors, so we really need to understand the vector, the human and the environment interface to see how the transmission will occur and where the outbreak will occur is so unpredictable. Another important, which Dr. Pragya also touched, is the Kyasnur forest disease, which is very typical to our country. 1955, it was localized to, single district, Shimoga of Karnataka. And subsequently, it spread to, had various districts of Karnataka reporting outbreak of Crimean Congo Kyasnur forest disease. Then neighboring state, Kerala, at the Karnataka-Kerala border, there are continuous outbreaks, spread to Goa, now Maharashtra at the Tri-Junction, then Tamil Nadu. So it is expanding its horizon, and definitely either we get a vaccine or we understand the virus, something needs to be done, and it has a very high mortality. Or we reduce, when we went into the history, people were traversing the forest. Some were doing it for fun. They wanted to see trekking in there, in those hills. Some were doing it, going for hunting. So these things, to decrease the interface between wildlife and human beings, is extremely important to, to avoid or prevent these outbreaks to occur. Then SARS-CoV-2, I won't talk much. Everybody has spoken about it, and it still continues. Lot of fear factor. I still remember the amount of fear it was in March 2020 when we were asked to go to Italy to get the stranded students. As a team lead, I was not supposed to be scared because I had to counsel others. But the state in Italy was similar. People did not have PPE kits. There were high mortality in Italy at that time. They did not have any facility to test the samples. So we had to bring the samples from there to India to test the samples to see what virus is circulating, whether we are introducing a new virus in the country. At that time, this is March 2020. So it has caused it did cause a lot of fear and psychosis. We were still dealing with SARS-CoV-2 when Mpox came in. Now, Mpox was a knee-jerk response, very reactive response, because it started in Europe, in MSM, male having sex with male. So, but as Dr. Pragya has already said, very quickly India did the preparedness, 35 labs were strengthened, and the diagnosis facilities were established so that the disease could curtail. Then came the hand, foot and mouth disease. Now you see what is reaction instead of response. There was this exaggerated or atypical presentation on foot and hands of children, and people reported as tomato fever. And the article was published in Lancet. So it was, again, there was a lot of thought process going in that it is a plant virus that is causing infection in humans. And so before even testing, the article was published. Subsequently, it tested, it turned out to be enterovirus. So that is a knee-jerk response. Instead, we should strengthen our surveillance, and hand, foot and mouth disease happens all over the country. Just because of atypical presentation, it was misreported. So Zika virus, Dr. Pragya has already spoken, so I will just skip this. Again, this was public health emergency of international concern. And why did it become a concern? It was because of microcephaly it caused in children. But it was associated with microcephaly that happens in children when the infection occurs in a pregnant mother. But still many, many questions remain unanswered as to when the infection occurred, how to diagnose it. We do not have any of those answers as of now. So the list is endless. I talk about Chandipura infection, which recently occurred in August 24 in Gujarat and Rajasthan, with case fatality rate of about 33%, 1/3 of people dying, and all these were children under age of 15 years. West Nile virus outbreak, leptospira, brucellosis, rabies, trichinella, scrub typhus, toxoplasmosis, list is endless. And soon we'll have, there are future threats which are happening in our neighboring countries. It's yellow fever, hantavirus, Rift Valley, which we have not reported in our country. Nothing stops from viruses to cross the boundaries. So threat is always there. And then there would be something new, and another virus will come in, and story shall continue. So there are big projections. There are 111 viral families. 25 of them can infect human currently. There are over 20,000 coronavirus that are circulating. We have had only 7. 1.6 million viral species can infect mammals and water birds. All these are threat and can become zoonotic. And there are about 7 lakh human viruses. So what are the lessons learned? Lessons learned are that zoonosis will continue to emerge. The basis is that we have to do early detection and establish the etiological diagnosis, and that is why a lot of attempt in now syndromic approach to diseases. Surveillance needs to be strengthened. And the most important thing that I learned was that joint orientation training courses, that you train medical and veterinary people and environmental people together for them to understand the importance and see what is required to make this place a better place to live, so that we leave a good environment for our kids. So what needs to be done is absolutely, as we've been hearing, of One Health approach is to work closely with the animal health sector and respect the coexistence of animals, humans, and environment. And this needs to be nurtured and protected in a very comprehensive approach. That is the One Health approach. As we've been talking about, there should be collaboration, not only intra-sectoral, but multi-sectoral, transdisciplinary, local, as Dr. Salunke put it, it has to be from the ground root. Because as CCHF outbreak occurred, when we went to the districts, the animal husbandry people did not know the health people at all. But because we were conducting this joint orientation training courses, we called them and we introduced them, so that because you cannot control an outbreak of CCHF without, knowing the animals which are involved, how is the trade taking place, unless we understand, it is very difficult to control such outbreaks. So One Health is validated, integrated, and holistic approach, and it has to work on these principles of 5 C's or 6 C's. We've already seen this slide, so I will just come to end of the presentation. And it is a holistic approach, a simple and powerful concept. However, it is extremely complex when it comes to implementation. So many, as Sir said, very rightly said, when we go to the ground, we realize how difficult it is to implement. You do it, I do it, she does it. These are the questions that we need to, answer on the ground. Who is going to fund? How is the funding going to flow? So these are some of the aspects that we need to resolve. And of course, it needs multi-sectoral, multi-institutional, multidisciplinary approach with the policy shift to, desectorize human, animal, plant, and ecosystem. So we need to do some basic research. That is very important. And of course, I have not spoken about AMR. That is the silent pandemic, which also need One Health approach, And my wholehearted respect to healthcare workers who have lost their lives. So in CCHF or in Nipah virus or in Corona, with those few words, emerging infections are only a flight away. And these microbes are much smarter than us. Their millennium is, our millennium is their 30 minutes. So they will outsmart us. They will change the species. And we have to keep abreast with their. So thank you so much. One should respect self, respect for human beings, respect for animals, respect for environment, and what COVID has taught us is your well-being is my well-being. Thank you so much for your time.
Thank you, Malaji. You are a very pleasant person, but some of the presentations, component of the presentation were fairly scary. I mean, let us really appreciate the kind of presentation in which she is bringing out really some of the great threats. So thank you very much, and I am pretty certain that all of you are quite thrilled with this presentation. I will be requesting organizers kindly share this presentation because this will be useful for us. Malaji, I will give you my email address.
Absolutely, sir.
Organizer भेजेंगे ना भेजेंगे आप भेज दीजिए.
जरूर, sir.
Okay. Are there any questions, queries that you want to ask to her? Very, very comprehensive presentation. Yes.
I did my PhD on crimean hemorrhagic fever in Moscow, 1970-75. I think only paper published in the Indian Journal of Medical Research in 1976. When the outbreak occurred in Gujarat, I started a lot of messages asking for me, have you any other publication? I have 4 publications on crimean hemorrhage. So I showed only seropositivity in few animals and human beings. I collected 1,200 sera from 6 medical colleges from Trivandrum to Delhi. So that was just an added information.
If not, organizers, we have exceeded a little bit of time, but I think these were very excellent presentations, and thank you again. Yes.
Excellent presentation. All had a great discussion about One Health program. We learn a lot and various epidemiological aspects and laboratory and safety and all issues discussed, all aspects of One Health discussed. So really thank you, ma'am. Thank you. Now I will request our chairperson to felicitate Dr. Mala Chhabra. Okay. Thank you, sir. Thank you, madam. Now I request sir to felicitate Dr. Pragya Yadav. Madam, please. Thank you, sir. Thank you, madam.